Hop on the Magic School Bus to learn about the cell membrane.
We are now at the cell membrane. All cells have a cell membrane. It is used to facilitate transportation of materials in and out of the cell. The phospholipid bilayer is composed of phospholipids arranged in 2 layers that determine what can go through it. The cell membrane is sometimes referred to as the fluid mosaic.
The extracellular side of the phospholipid bilayer faces the cell's environment, while the intracellular side faces the inside of the cell.
This is the phospholipid. The phospholipid bilayer is amphithetic with hydrophilic heads and hydrophobic tails. Hydrophilic directly translates to water-loving. Hydrophilic molecules are polar. This is the head of the molecule you see. The tail is hydrophobic, directly translating to water-fearing. Hydrophobic molecules are nonpolar, and these are the 2 tails that you see.
This is an integral protein. These proteins are embedded within the bilayer. They can assist in transporting and channeling molecules through the bilayer, as long as they're transmembrane. They can also act as cell receptors or help with cell adhesion. Keep in mind, the integral monotopic proteins are permanently attached to only one side of the bilayer.
These are transmembrane proteins, proteins that span the entire lipid bilayer. They assist in transporting and channeling molecules through the bilayer. They are integral proteins, but not all integral proteins are transmembrane proteins.
We have reached the peripheral proteins. These are proteins that only temporarily attach to the cell membrane and help with cell signaling.
We are now at the glycolipid, which can act as receptors, help in cell adhesion and dissociation, and help with signal transduction.
This is a glycoprotein. A glycoprotein is a molecule consisting of protein and carbohydrate chains, help in protein folding, protein targeting, receptor function, cell-cell recognition, protein stability, signaling, and more.
Cholesterol are lipids that change the fluidity, thickness, compressibility, and water penetration of the bilayer, and they're located in between phospholipids.
There are 4 types of junctions. Tight junctions seal adjacent epithelial cells in a narrow band just beneath their surface, limiting the passage of molecules and ions between the cell and block the movement of integral proteins between apical and basilateral surfaces of the cell. Next, adherens provide strong mechanical attachments between adjacent cells. For an example, they hold cardiac muscle cells tightly together as it expands and contracts. Next, gap junctions are intracellular channels that permit free passage between the cells of ions and small molecules. And lastly, desmosomes junctions are localized patches that hold 2 cells tightly together, and they are attached to intermediate filaments of keratin in the cytoplasm.
Enzymes are sometimes membrane-bound proteins and also can be bound to the membrane. They catalyze biochemical reactions. This structure allows them to uncover the enzyme mechanism to permit hydrophilic and hydrophobic molecules to interact without leaving the natural environment. And in bacteria, they are key steps in assembling the glycojugenates, which help bacteria survive. One part sticks outside the cell membrane into the watery cytoplasm of the cell, which captures the hydrophilic molecule, which the second part of the helix pokes halfway into the membrane bilayer, where it binds to the hydrophobic molecule polyprenyl phosphate. PGLC brings these 2 molecules together in the third part of the enzyme, which is located on the membrane surface and where the active site is. The active site links structures together.
The membrane is selectively permeable, so it decides what comes into the cell. The transport system prevents random substances from simply entering and exiting the cell. Transportation occurs in a few ways, such as passive diffusion and facilitated diffusion. Passive transport is where molecules simply dissolve into the phospholipid bilayer and diffuse across it, or when they go through protein channels. Meanwhile, active transport is when energy is used, such as with protein pumps or carrier proteins. This is used to transport larger molecules. Recognition proteins are proteins in the plasma membrane that provide a fingerprint on the outside-facing surface of the cell and are essentially recognition to other cells. A recognition protein is made up of a glycoprotein that distinguishes among cell types and a carbohydrate bonded with a protein that extends outward from the cell and is used to identify other cells and the outside world. For example, an individual will have certain recognition proteins which are on the surface to identify the cell as belonging to that individual's body. Those cells are ignored by the immune system, while others identified as not belonging to the individual will be attacked. Signal transduction is used in directly regulating enzyme activities as it triggers the formation of secondary messengers and ultimately controls gene expression. Signal transduction triggers many responses, which may include changing the metabolism of the cell, receiving the signal, or changing the gene expression, either within the nucleus of the cell or just in the cell. The steps of cell signaling start with reception, where a cell detects a signaling molecule from the outside of the cell, and then transduction, when the signaling molecule binds to the receptor and it changes the receptor protein. And lastly, response, where the signal will trigger a specific cellular response. Adding or removing phosphate groups in this can result in the activation of proteins, which then allows the enzyme to transfer phosphate groups from ATP to a protein called protein kinase, which can result in a phosphorylation cascade. Small, non-protein water-soluble molecules, or ions, called secondary messengers, can also send signals received by receptors on the cell's surface to target molecules in the cytoplasm or nucleus. Membrane proteins in anchorage and attachment represent one of the largest and most important classes of proteins. Membrane proteins anchorage and attachment refers to the process of providing attachment sites for the cells within a tissue, and this helps in regenerating tissues by giving stability and guidance for cell growth. Anchorage and attachment is important for the cytoskeleton network, protein location, and upholding of cells. Other proteins also bind to molecules inside and outside of the cell, which maintains the structure of the cell.